Hi, I'm Toto Olivera from the University of Utah, and I'd like to tell you about the research we've done with a group of snails called cone snails. And so what I'm going to do is first give you a history of how we got started with this particular research project. So cone snails, it turns out, are predatory animals. And if you look at predators, you can think about them as either primarily using a mechanical strategy, such as the great white shark. And so predators like this have a lot of speed, a lot of weaponry, so the teeth of a great white shark, for example. But there's a lot of variation in whether an animal will also use chemicals as part of its strategy for capturing its prey. And what I'm going to show you is that cone snails are at the very end of the spectrum with regard to the chemical strategies that they use. They basically don't have anything going for them mechanically. They're slow moving. They really are not going to be able to chase prey that's faster. They don't really have any kind of armamentarium or weaponry to attack their prey. And of course, there are animals in between, like the Komodo dragon, which does have big teeth. It's able to bite its prey, but it also uses a venom as part of its strategy for capturing prey. So the cone snails are really chemical specialists as predators. And what I'm going to show you is that there's a diversity of different cone snails. It's actually a very successful group of animals, 700 different species. And each cone snail is fairly specialized as to what it's going to use as its prey. And so if we look at different cone snail species, what we find is that some snails, such as this one in the center here, will only eat fish. And so it essentially ignores all other animals except fish. In contrast, this species over here will eat other snails. And this species up on top, which is called the imperial cone, because it has a little crown on its top, that will primarily eat marine worms, a certain type of marine worm called a fireworm. So all of these cone snails will use their venom in order to capture prey. And what you're going to see is that the venom is very, very effective when it comes to prey capture. So how did we get started working on this group of animals? It really was early in my career. I went back to the Philippines, and while in the Philippines, we ended up with a lab that had no equipment whatsoever. And I had done a postdoc at Stanford University, where we had every piece of equipment we might want. So it was sort of a shock to find that I couldn't do DNA metabolism, DNA synthesis, which is what I had been working on. And so we had to find something else to do. And because I had collected shells as a kid, we knew that this snail in the center here, Conus geographus, that this snail was capable of killing people. And so in the absence of medical intervention, there's a 70% fatality rate. So this is a very potent venom. 70% of the people who get stung die. And so we wanted to understand why. So that was our initial goal. I thought this would be a short-term project and that after a while, I could return to DNA replication. So we decided to purify the components of this venom, which were responsible for human fatality. So here's a problem. If you want to understand what kills people in a venom, First of all, you have to have enough of the venom to purify out the component you want. And that's not a problem because Conus geographus is not a rare shell in the Philippines, so we can get lots of snails. But what you really need is an assay. And it's not completely clear what kind of assay you should use if what you're interested in is what kills people. So you have to find an assay, of course, that's ethically admissible. And so what we did was we used an assay based on the medical reports in the literature that said Conus geographus killed people because they got paralyzed. After a while, their diaphragm was paralyzed. They couldn't breathe. And so what we decided to do was to use the assay shown in the next slide. I'm very proud of this slide because it really shows all the equipment we had in the lab at the time. And so what we would do was we would take a mouse, which the med school class at the University of the Philippines had used for nutrition experiments, so we could take the controls. We'd put the mouse upside down on a wire screen, as shown here, and we would inject fractions of the venom. And if the venom were paralytic, then the mice, which normally could hang on to this wire screen essentially indefinitely, as their little paws got paralyzed, they wouldn't be able to hang on anymore and then fall. And we call that the falling time. And so that was our assay. What we did was we'd inject essentially components of the venom into a mouse and observe whether or not they had, whether it caused them to fall. And we measured the falling time. And so you might think this is incredibly crude and very unquantitative, but surprisingly, once you measure the falling time, what we found was that the falling time followed what we call Michaelis-Menten kinetics. That means that if you plot the dose, the reciprocal of the dose, which is shown here, versus the falling time, which is really the reciprocal of the velocity, what you get is a straight line plot, which Michaelis and Menten, famous enzyme kineticists, had already developed as a theory. And so, surprisingly, the falling time is a very quantitative assay, and this allowed us to quantitate how much paralytic activity there was. And so, using this very simple assay, we were able to purify the two components in the venom of this snail that caused paralysis. And those two are shown here. And so on top is the first peptide that was purified. Today, this is called alpha-conotoxin G1. And this acts very much like cobra toxin or alpha-bungarotoxin. So when you get bitten by a snake related to cobras, the stuff that kills you is a toxin that basically prevents communication between nerve and muscle. And so cobra toxin and alpha-bungarotoxin are examples of that. It turns out that this peptide that we purified from Conus geographus venom acts by exactly the same mechanism. And in fact, this peptide competes for binding to the same site as cobra toxin and alpha-bungarotoxin. The difference is that snakes make a small protein, 60 to 80 amino acids in length, while the snail makes an extremely small peptide, 13 amino acids, and that was really a surprise at the time. The second component we isolated, which was also paralytic, is shown here, mu-conotoxin, which is 22 amino acids in length. And what you can see is that the peptide has 3 disulfide cross-links. The cysteine residues are cross-linked to each other. So this is a small disulfide cross-linked peptide. And it turns out it acts by a different mechanism to cause paralysis. So I think many of you know that in Japan, one of the great delicacies that people like to eat is pufferfish, fugu. And so if you want to eat pufferfish, in general, you have to go to a fairly fancy restaurant, pay lots of money. And this isn't because pufferfish are rare, because in fact, pufferfish are pretty common, But because if the chef isn't trained properly, then the customers begin to drop dead. And the reason they do is because in pufferfish, in certain tissues, at certain times of the year, there's an alkaloid-like molecule called tetrodotoxin, which wipes out action potentials by inhibiting voltage-gated sodium channels. Well, it turns out that this peptide, although it's chemically completely unrelated to tetrodotoxin, acts by exactly the same mechanism. And in fact, this peptide binds to the same site as tetrodotoxin. So by isolating these two peptides, we were in fact able to address the question we originally wanted to, which is, why does the venom of Conus geographus cause lethality? And the answer is that when you get stung by the snail, it's the equivalent of being bitten by a cobra and eating a lethal dose of pufferfish at the same time. And so you can see that that's not good for you, and that would account for the 70% rate of lethality. So we answered the question we wanted to, and so you might ask, well, that was a long time ago. Why are you still working on cone snails today? And the answer is that very often, when you're focused on a certain question, you stumble on something that's more interesting along the way. And in our case, I'm going to show you exactly how we stumbled on something unexpected. So here is how we used to purify Conus geographus venom. So what we would do is we would essentially take the venom and size it, separate it according to size. And so in fraction B, those are the peptides that are about 20 amino acids in length. And in fraction D, these are the peptides that are about 10 to 15 amino acids in length. And if you take these size fractions, and now you put it on a more sophisticated separation device called an HPLC column, high performance liquid chromatography, what you can see is that fraction B separates into maybe 40 or 50 different components. Each peak here is in fact a peptide that's about 20 amino acids in length. So these are all found in the venom of Conus geographus. And the labeled peaks are the peaks that would score on the falling time assay. So they cause paralysis, and these are variations of the peptide that's related to tetrodotoxin in its biological activity. So what you can see is that most of the peaks are not labeled, which meant that when we injected them intraperitoneally into a mouse and put the mouse up on a wire screen, they didn't do anything as far as we could tell. And so for that reason, we pretty much ignore them. However, a young undergraduate of the University of Utah, Craig Clark, came up into the lab and he said, you know, you guys are doing the assay all wrong. You should be injecting directly into the central nervous system, directly into the brain. And I wasn't persuaded. I thought this wasn't such a good idea. What did we learn? And I tried to dissuade him. But I really feel that the reason why the most creative research is done at universities is that the students do what they want. They don't follow what their professor advises. So fortunately for us, Craig went and essentially took all these fractions, and instead of injecting them intraperitoneally, what he did was he injected them directly into the central nervous system. And this is the same chromatogram that I showed last time, but now instead of just the paralytic peptides being active, which eluded at the beginning, now all of those previously inactive venom components caused a phenotype when injected into mice. And so what you can see is that in addition to the paralytic peptides, the next peptide over caused mice to jump and twist as they're jumping. The next peptide caused mice to become uncoordinated in their movements. And then the major peak, which previously had shown no phenotype, now put mice to sleep. And the mice would be asleep for 12 to 24 hours, and they'd wake up, and they were perfectly fine. But that was only true if the mice were under 3 weeks of age. In contrast, older mice, when this same peptide was injected, would climb the sides of their cages, run from corner to corner. And so you got a climbing phenotype instead of a sleeping phenotype. The next peptide over made them drag their back legs. The next peptide Craig called the circular peptide because the mice would run around in circles. The next peptide made them swing their heads back and forth. The next peptide made them kick on their back and scratch. And then there was a peptide that caused trembling, a peptide that caused scratching, a peptide that caused convulsions. And so you can see the remarkable result was that almost every peptide caused a behavioral phenotype, and the effect of each peptide was different. And so this made us realize for the first time that the venom of these snails was not a mixture of a few paralytic toxins, but was this incredibly complex pharmacological mixture with a lot of pharmacological diversity. And that's the reason we're still working on cone snails today. So while Craig was doing his work, he also was great at recruiting other undergraduates to join the project. And so each undergraduate could choose any cone snail they wanted and follow any activity they wanted. And this young guy, Michael McIntosh, right out of high school, decided to purify a component from Conus magus venom, so that's the magician's cone. And what you see here is the peak that he decided to purify, which caused shaking. And so he called this the shaker peptide. And the mice would have characteristic tremors after injection with this peptide that were very, very reproducible. And so the job of these undergraduates was they had to purify the peptide from the venom until we could analyze what the sequence of the peptide was. Then we could chemically synthesize the peptide, and then we could figure out what that peptide really did. And so that's what Michael did. And the reason I'm telling you this story many decades later is because, in fact, that peptide today is an approved drug for intractable pain. So here's the sequence that Michael determined, and the peptide was synthesized by our collaborator, Jean Rivier, at the Salk Institute. And today, it's an approved drug for intractable pain. We call this peptide omega conotoxin M7A. Well, if you're going to sell something as a drug, omega conotoxin is not exactly the best name. And so, in fact, today, this drug is called Prialt, which is primary alternative to morphine. And so what I'd like to show you is a retrospective summary of how Prialt acts. So first of all, why did the snail evolve this peptide? This snail hunts fish. And so the fish prey, of course, have a nervous system in which action potentials are fired, and those action potentials have to cross the neuromuscular junction, and only then can a muscle contract. And so this is a close-up of an action potential which crosses the neuromuscular junction. And as the action potential reaches the nerve ending, as shown here, what happens is that voltage-gated calcium channels, which are in fact shown in green, open up in response to the action potential. They let calcium in, and the entry of calcium into the presynaptic terminus causes the fusion of vesicles, and so that fusion causes the release of the neurotransmitter, which is acetylcholine, as you can see. And now the neurotransmitter crosses the synapse and binds to its target on the muscle side of the synapse, and as a result, channels open, and the action potential is therefore transmitted across the neuromuscular junction, and you get a faithful transmission of the action potential across the neuromuscular synapse. So what does this peptide do to interfere with this normal physiological process? This peptide hones in very specifically to the voltage-gated calcium channels, blocks them. And so now, in the presence of the peptide, even when an action potential reaches the nerve ending, there is no calcium that enters the presynaptic terminus. The presynaptic terminus, therefore, does not release the neurotransmitter. And as a result, the action potential is stuck at the nerve ending, does not cross the neuromuscular synapse. And therefore, in essence, the muscles never can contract. So this is a paralytic toxin. How do you make a paralytic toxin into a drug? Well, it turns out that if you look at mammals, like ourselves or like mice, essentially the same normal physiological process occurs. And so what you have here is pre-alt, but there's a difference. The calcium channels are not green, they're red, okay? They're a different subtype. And it turns out that those calcium channels are completely resistant to this peptide. And so if you inject this venom component into a mammal, it doesn't cause paralysis because you have a different type of calcium channel that's present at the neuromuscular junction. However, we do, in fact, have green calcium channels. Mice, mammals, have green calcium channels, but they're in different places. One of the places that's important is they're in the pain circuitry. And so when you feel pain, what happens is the pain fiber keeps on firing action potentials. That's the signal for pain. But for us to perceive the pain, that action potential has to cross a synapse. And so now, if pre-alt is present, then what you find is that, again, the action potential reaches the nerve ending, but now no neurotransmitter is released, and so the signal is stuck at the synapse. And what this means is that even though the pain fiber is signaling pain by firing constantly, what's happening is we do not perceive the pain because essentially that synapse is blocked. The signal never reaches our spinal cord, never reaches our brain. And therefore, even though we have very severe pain, we are, in fact, pain-free in our central nervous system. And that is the basis for how this peptide became a drug. And so you can see that what is used as a toxin for paralysis, on the one hand, can be used as a drug for severe pain in humans. And that's why it's now an approved drug when patients become tolerant to morphine and morphine no longer helps them. It's interesting to note that the biotech company that originally developed this peptide did a lot of structure function work. They essentially changed every amino acid in the peptide to try to make it better for its therapeutic purposes. But in the end, they went with exactly what the snails make. And so the commercial product is identical to the natural product, except that it's chemically synthesized. There's not a single functional group that's any different. So what do the snails use these peptides for? And what I'm going to show you is a video of a snail that's attacking its prey, fish. And so what the snail does is when it sniffs a fish with its siphon, it extends its highly distensible proboscis towards the fish, as you can see here. And now this proboscis can be targeted, and this particular species looks for the lateral line of the fish. And when the snail touches the skin of a fish, this particular species, what happens is that from the tip of the proboscis, which is shown here, out comes a harpoon-like tooth, which is both harpoon and hypodermic needle. And what you can see is that the venom will flow from the proboscis through this disposable harpoon-like tooth and finally end up coming out at this end. As you can see, there's a hole at the end, and this is barbed. And it's really shaped like a harpoon. And so when this comes out of the proboscis, and here's a harpoon from a different species, so every cone snail species has its own harpoon designs, if you will. But here's what happens. when it finally brings out the harpoon-like tooth. It tethers the fish and simultaneously is injecting venom. And so now the fish is tethered. It's brought towards the false mouth of the snail. And what the snail does is it completely engulfs the fish. So in about an hour and a half, this snail will regurgitate the scales and bones of the fish and the one harpoon that it used for injecting the venom. And so, in fact, this harpoon-like tooth is the equivalent of a disposable hypodermic needle. And so about 50 million years ago, these snails evolved this drug delivery device, and ever since then, they've been more and more successful. What I'd like you to notice in this particular video is that the fish has very, very stiff fins. I think you'll agree you've never seen such a stiff fish in your life. So all the snails have the same venom apparatus, all cone snails, no matter what they hunt. They have a quiver-like organ, which has between 50 and 100 harpoons in various stages of assembly. And when they want to go hunting, what they do is they move one of the harpoons to the proboscis, and they use it for injecting the venom, but they only use it once, and then they throw it out. The venom is actually made in this long duct, convoluted duct shown here, and is pushed out by the bulb, the muscular bulb, which kind of acts like a medicine dropper bulb and pushes the venom out through the proboscis, through the harpoon, and into the envenomated prey. So what we did for a fish hunting cone snail was to simply isolate all venom components that paralyze a fish. And to our surprise, there are many components that by themselves can isolate a fish. So here are 5 that are found in the same venom. As you can see, some of them seem to be related to each other, some not. But they're all small peptides between 13 and 30 amino acids. And it turns out that they all act at the junction between the motor axon and the muscle, at the neuromuscular junction. And so, as I showed you in the video earlier, there's some peptides that block calcium channels, so we call those omega conotoxins. It turns out that there's another group of peptides that block the nicotinic acetylcholine receptor, the postsynaptic receptor. Included among these are alpha conotoxins. And in addition to blocking the presynaptic terminus and the postsynaptic terminus, the snails also block the action potentials on the muscle membrane, and those are the mu conotoxins. So what's going on here? Any one of these is sufficient to paralyze a fish, and yet the snail is making typically 5 different components. And what this means is that the snails discovered the principle of combination drug therapy many millions of years ago. And as far as we can tell, they never just use a single pharmacological agent to achieve a particular physiological endpoint. They always use a combination. And so we call groups of toxins that act together in a synergistic way a cabal, because cabals are secret societies out to overthrow existing authority. And so we call this group of peptides that blocks neuromuscular transmission the motor cabal. And so that shows you the toxins and the pharmacological site that each toxin in the motor cabal targets. So we're pretty proud of ourselves for having figured this out, that what each peptide does. But in fact, there's a problem. If you take any one of these toxins and inject it into a fish, or even if you took all of them in combination and injected massive amounts of these peptides into a fish, it will take you about 20 minutes before you can paralyze a fish. And yet, you saw from the video that it actually took much faster than that before the fish was immobilized. Furthermore, when you take these peptides and inject it into a fish, the fish looks as shown in the lower cartoon. It's flaccid. All the fins are relaxed. And that's because you block neuromuscular junctions. But of course, when we looked at the fish when it was actually envenomated, the fish had very, very stiff fins. what's going on here? It turns out there's a second cabal. In addition to the motor cabal, there's a lightning strike cabal. And how that acts is that there are two principal components called kappa toxins and delta toxins, and two examples of those are shown here. And how these behave is shown in the animation. And so what you have is, again, a fish and action potentials. And of course, action potentials are carried by axons. And so an action potential is initiated when sodium channels, shown in blue, open, and is terminated when the sodium channels go into an inactivated state. And so as the sodium channel is activated by a depolarization on the membrane, it opens transiently, goes into an inactivated state, as shown here, and then potassium channels open, let potassium out, and that terminates the action potential. And so action potentials are transient because of both sodium channel inactivation and potassium channels opening. How does the snail subvert this? Well, what I'm going to show you is that if a delta toxin binds to the voltage-gated sodium channel, it opens reasonably normally, but when the delta toxin is bound, it never inactivates. It remains open. If you now combine that with the kappa toxin that blocks potassium channels, what happens is that you've inhibited both mechanisms to terminate an action potential. And so that part of the membrane remains depolarized. It remains in a state where it generates action potentials. And so what happens is from the site of injection, you have an electrical storm across the entire organism. And that's why you get this very, very rapid paralysis. So the equivalent of this pharmacological cocktail is taking a powerful taser and zapping the fish at the injection site. It's really equivalent to that, except that the snails have figured out how to do it pharmacologically. And so we call this the lightning strike cabal. So in fact, to capture prey turns out to be more complex than you might have expected. Two cabals are required. And each component of the cabal, and I haven't had time to go into the mechanism of every component, has a particular target, a molecular target, and the molecular targets of the lightning strike cabal are different from the components of the motor cabal. But they're all ion channels. And so what you see in the summary are the various ion channels that are in fact being targeted. So the strategy of the snail is, first of all, the lightning strike cabal, those toxins don't have to spread through the body of the prey. They can act right at the site of injection. All they have to do is glomp onto the axons in the neighborhood of the injection site. Now, the components of the motor cabal enter the circulation, get to the neuromuscular junctions, and cause a final irreversible block of neuromuscular transmission. So you might say, well, if you have a lightning strike cabal that acts so fast, what do you need a motor cabal for? Why do these snails, why have they evolved the motor cabal? And what I'm going to show you is that in this video, the fastest immobilization we've ever seen for a fish hunting cone snail. And so here's, you can see it's just starting to stick its proboscis out. And what you're going to see is how rapidly the lightning strike cabal acts. And so here's the snail sticking out its proboscis, and you're going to see that's the lightning strike cabal. This is real time, so you can see how fast it is. The reason I'm showing you this particular video is something unexpected is going to happen. And so as the snail is engulfing the fish in this case, what you're going to see is that, in fact, the fish is going to recover. And so if you watch the video, you'll see there's the fish. It's recovered. In this case, of course, it's too late. The fish is already engulfed in the mouth of the snail. But we have seen these snails harpoon a fish, and as they're reeling in the fish, if the fish is a big fish and it hasn't been harpooned so completely, the fish recovers. And before the snail can reel it into its mouth, the fish is able to break away and swim away. And so it can't be reeled in. When that happens, the snail always does a search of the neighborhood because it's expecting to find a paralyzed fish. And so what this says is that in the real world, there's enough advantage in having two cabals so that the selective pressure to get two groups of toxins is enough. And so in the real world, to be an effective predator, fish hunting cone snail, you have to have two cabals if you're using this particular strategy. And that, of course, is much more sophisticated than we certainly predicted at the beginning of these studies. And so, in summary, we think that the snails are using a strategy called constellation pharmacology, where the venom peptides are organized into cabals. They act on targets that we call constellations of functionally linked ion channels and receptors. And a cabal acting on its target constellation has a dramatic physiological endpoint. But the principle that I'd like to emphasize is that every venom peptide has been evolved to target a very specific molecular target in a particular physiological context. However, the toxins in cone snails are used for more than prey capture. Predators are attacking them, as shown in this cartoon, and they will use their venoms defensively. Certainly people who've gotten stung, it's not because the snail thought you were a fish, it's because you've done something stupid to make the snail think you're a predator, and that's why they sting you. In addition, they use their venoms for competitive interactions. It's very difficult to document this in the field, but in an aquarium, we've seen a small species of cone snail attack a worm, and this species, they all chomp on the worm together, and we saw another species of cone snail get to the worm later, and when it harpooned the worm with its venom, suddenly we saw all of the small guys release the worm. And so clearly there's something in the venom of the big guy that deters competitors. And so the reason these venoms are so complex is partly because they use a cabal strategy, so they always use combinations for every physiological purpose. But then in addition, they target multiple animals. They use their venom to capture prey, to defend against predators, and for competitive interactions. And so this is a summary of the biology of why we have species with very, very complex venoms. And what I'd like to acknowledge is that this work was done as a part of a big collaboration and with the support of the GM Institute at NIH. Thank you very much. Next time, I'll not talk just about fish hunting cone snails, but about the biodiversity and evolution of cone snails and the molecular basis for that. Thank you.